All right, nerds. So we're going to look at today at what a carbohydrate is essentially and how we differentiate between plant and animal carbohydrates. All right. So basically, organic materials, they're found at the scenes of crime. So we need to use, we need to be able to differentiate between both animal and plant as well as specific animals and specific plants so we can determine what was found at the scene of the crime or if something was there and so forth. Um, in particular, this is quite difficult, but also very important in arson cases where trace evidence is very difficult, and organic materials tend to, when they're combusting, decay the same. Whether they're like at the end, your plant and your animals, they're tough to tell apart, but you can chemically, you can usually do it. All right, so you also want to be able to explain the chemistry behind several different types of saccharides or carbohydrates. So what is a carbohydrate? Well, basically, it's an organic compound consisting of only carbon, hydrogen, and oxygen, and we call them sugars. They have this general formula here, where you've got CX on the outside with a H2O, and Y. And the X and the Y can be different numbers, or they are different numbers. They can be the same, but they're not necessarily related. So glucose is the simplest, which we can see as a C6H12O6, or we could draw it as C6H2O6. So basically, a carbohydrate is a repeating sugar molecule, or a sugar molecule on it. Now, what is a sugar molecule? We'll look at that in a second. They're classified by their monomer units. Okay, so the monomer units are the individual sugar rings. And we use them. They're really important, and they're important for the source of energy, structure and supportive components of cells, as well as for growth and development. And we can classify them into three main groups. We've got monosaccharides, we've got disaccharides, and we've got polysaccharides, which are much larger polymers. So here's a little summary of what we've got. So these are our monosaccharides. So we've got glucose, fructose, galactose. There are more than this. Disaccharides. These are the ones that we tend to think of as food. So you've got sucrose, lactose, and maltose. Maltose is the one which is glucose. And then there's the other aspects of food, as well as non-foods. Cellulose doesn't tend to be food for us. It's fiber, but it's not the same way this is food. We've got starch, glycogen, and cellulose. And glycogen is special because that one's only found in animals. All right, so what are the monosaccharides? They're the simplest carbohydrates. They cannot be broken down, so they cannot be hydrolyzed. Hydrolyzed, we'll go more into hydrolysis later on, but hydrolyzed means to split. That's what lysis means, split with water. We'll go more into that in a lesson or two. So they can't be split into smaller sugars. Monosaccharides with 5 carbons are called pentoses, 6 carbons are called hexoses, and you'll see that it's the ose at the end, which is what makes it, that's the one that the suffix which denotes that it's a sugar. So the common pentose is ribose, and this is the basic unit for deoxyribonucleic acid, or ribonucleic acid for both of them. The common hexoses we see are glucose, fructose, and galactose, and they're all isomers of, well, obviously these numbers should be in subscript, of C6H12 and O6. Now, glucose we get from photosynthesis in plants. Okay, so that's the source there. It exists in the two isomers, which we'll have a look at in a second. We'll have diagrams of these in a minute, of alpha glucose and beta glucose. Now, in alpha glucose, the OH group of C1 is always opposite to the OH group, while in, and you'll see what I mean in a second, to the C2OH group, while in beta glucose, they're on the same side. It also applies when we're naming fructose and galactose. So here we have alpha galactose, and we see there's an OH over here, and there's an OH there, and they're on the same side. Okay, so that's obviously cut off. That's a pen. That's an H there as well. So we've got an H there at the same time. That's alpha. And on beta, they are on opposite sides. And this makes a difference when we start talking about our polymers, but we'll get to that in a minute. Galactose here, we see that this is actually a beta galactose. All right. And fructose, well, actually, here we look at this one here. You see that is also a beta, but it's an isomer, so you can have both ones. So what is a disaccharide? It's pretty simple. It's just two of them joined together. So if a carbohydrate can be hydrolyzed into two simpler sugars, these is at least a disaccharide and might be a polysaccharide. Now, hydrolysis is the opposite of condensation, and we do it through gently heating them with water and acid. And it's always a dilute acid, and the acid is there to catalyze the reaction. So a disaccharide, it's when two monosaccharides combine and they go through they form a glycoside bond via a condensation reaction. We'll have a picture of this in a sec. See if we can pull on it now. So you can see, yeah, cool. So we've got a picture coming up. So they form a glycosidic bond or a glycoside bond, which is this one here. Sorry, that's the whole thing, through a condensation reaction. So the carbon from each monosaccharide that reacts to form the glycoside bond is known as the anomeric carbon. That's going to become a very important word in the next lesson. And it is C1 for glucose and galactose and C2 for fructose. We'll have a look at this in a diagram in a second. All disaccharides are soluble in water and have a sweet taste. The common disaccharides that we look at are sucrose, maltose, and lactose, and they're all isomers of C12H22, C011, or C12H2O. That should be a 2 there. No, it shouldn't. No, it shouldn't. That standard formula doesn't exist. My bad. Move on. All right. So here we've got two glucoses, or it could be any of them really, joined together. So two monosaccharides joined together to form this disaccharide plus water, because it's a condensation reaction. So maltose is formed with two alpha glucoses. Lactose is formed with a glucose and a galactose. And sucrose is formed with an alpha glucose and a beta fructose. And let's have a look. So we'll just quickly clear the ink. What we have here is we see here comes your glucose and your fructose together. This bonds together. This water bonds and separates over here, and it makes a condensation polymer reaction. So you have sucrose and water. Over here you've got glucose and galactose, and they join to form lactose plus water. And over here we've got glucose plus glucose, and this gives us maltose plus water. And that's pretty straightforward. Now the polysaccharides. These are really common, and these are the sorts of things that a forensic scientist will spend most of their time trying to identify. So these are long chains of carbohydrates that consist of like hundreds and thousands of monosaccharides. And they're formed via a condensation reaction, much the same as a disaccharide, but just continue the chain reaction along. And the most common ones are cellulose and starch, which are found in plants, and glycogen in animals. They're not the only ones, they're just the ones we're going to talk about. Cellulose, starch, and glycogen all use glucose as their monomer, which is why we're focusing on them, because they're the simplest ones. They just use glucose over and over. Now, so they all have the general formula, so it's using glucose of this, and N being however many chains monomers there are. So the glucose monomers form glycosidic bonds or glycoside bonds with each other. And the glycoside bond, by the way, is just COC. That's the glycoside bond. So that one there is your glycosidic bond. We'll talk more about that next lesson. And they eliminate water molecules. And this is what they look like. So you've got glycogen is this long branching chain, and starch exists in two forms. It exists as amylose, which is the one that reacts very well with iodine, and it exists as amylopectin, and we've got the ratios there in a second. And cellulose exists as these fibers, and they're long and straight. There's very little to no branching at all. So starch is made from 20% amylose, 80% amylopectin, and the amylose itself is actually soluble in water, while the amylopectin is not soluble in water. Cellulose is glucose, it's unbranched, and it also isn't soluble in water, it's straight, it makes these very straight chains. Glycogen is an animal starch, and it's branched. It's also not soluble in water. Okay, so now we're going to look at the 3 carbohydrates. We're going to focus on these 3, but in a bit more detail. So, cellulose. It's a condensation polymer of beta-glucose. So you can see you get this, see how you get the chains, the glycosidic bonds. They're up, they're down, and that allows it to pack really tightly. So due to the structure, it flips every monomer, flips the beta linkage, flips every monomer, and it's very linear. Has many parallel fibers, and they're held together through hydrogen bonding, so between the O's and the H's in between, so you have another group along here, and the H's and the O's hydrogen bond together. It's insoluble in water, and it's very difficult to decompose with chemicals. For example, we can't digest it, but cows which have the ruminant stomach do. But it's actually, most animals can't digest cellulose. So it makes up the main component of biomass, and it's there as plant cell walls. And we synthesize, well, we don't synthesize, it's synthesized in plants from glucose that's come from photosynthesis. So let's have a look at starch. Starch is a bit more interesting, I think. It's 2 types of condensation polymers made from alpha glucose, and that's amylose, and amylopectin. It's amylose. Amylase is a protein which breaks it down. We'll talk more about amylase later. So starch is in fact a complex structure, and it's got these amylose helices entangled with the branched amylopectin. So amylose isn't straight, but it's because of the alpha glucose. There's no flipping, so it's not linear. It sort of curls around itself. It's stored in cytoplasm as plant cells, and the plants use it for energy. They use cellulose for structure, starch for energy. And this is what it looks like. And it's this over and over. And because it's this, it just sort of makes this sort of helical structure as it goes. It just curls around itself. So that's amylose. It's about 20% of starches, but it varies within species, obviously. It's between 60 to 300 units long. And it's unbranched and helical in structure. It's soluble in water. Amylopectin, on the other hand, is branched. And it's the rest of it. It's about 80% of it. But again, it varies. It's much larger. It's like 300 to 6,000 glucose units. So what you actually see happening is when your amylose is made, it stops at about 300. But if it keeps going, then it stops being able to be this sort of helical, straight molecule. It starts to branch, and that's when. So if this is made longer than it was going to be, that's when it becomes amylopectin. And so you've got like the alpha 1,4 glycosidic bonds and some alpha 1,6 glycosidic bonds. So that's up here. So you've got that's a 1 and that's a 6 right there. So yeah. The branching occurs every 12 to 25 monomers, and through the alpha 6, that's this one here, sorry, the 1,6 bond, and the glucose condenses with 3 other glucose monomers instead of 2. So you see this one here would be attached to 2, so it's like 1, 2, whereas this one's attached to 1, 2, 3. And it's also insoluble, that one's insoluble in water. And then we've got glycogen. Now glycogen is what exists in animals, and you can see that it's quite this very complex structure. It's, again, like all the others, it's a condensation polymer from alpha glucose monomers. Very similar structure to amylopectin, but the branching is much more frequent and much shorter. So it has a lower molecular weight than amyloid pectin, so it's smaller. It's synthesized by animals from glucose, and we use it as a short-term energy storage. So in humans, it's stored in the liver, skeletal muscles, and small amounts in the kidneys and blood. We produce glucagon in the hormone, and this is what breaks it down into glucose. And this is regulated by the blood sugar levels. Muscle, it's used for muscle contraction as well. So glycogen breaks down. Glycogen is breaking down by enzymes, and this allows us to use glucose as energy in our muscles. So it's the frequent branching that really makes it easy to be broken down. If it was a straight chain, a very straight long chain, it would be hard to break it down, well, harder to break it down, bit by bit. Because it's got these little branched off bits, you can just snap bits off the large molecule. And that's it. There was a lot there. Hope you took it in, and we'll see you in class.